good morning, ladies and gentlemen. First of all, I would like to thank the organizers for giving me the opportunity to speak at this conference. Earlier, we put forward the hypothesis on the existence of a novel regulatory network, which we called transcriptional interference network. So this hypothesis is based on the interaction of the neighboring genes through the collision of their transcriptional machineries and the transcriptionally overlapping regions. So we proposed that these kind of interactions form a system-level self-regulatory network and result in a strictly controlled progression of the on and off states of the genes. We used the herpes virus model for these investigations to show that our hypothesis works in nature. So we originally assumed or proposed three different basic types of interactions. One of them is called as a waterfall model, which is represents a unidirectional interactions. Typically, two parallel overlapping genes can exhibit this kind of interaction, which means that the transcriptional machineries or the RNA polymerase of the upstream gene will somehow interact with the downstream gene. This interaction is typically the dislocation of the transcriptional initiation complex. The second basic type of interactions we called as a seesaw model, which is a bidirectional interaction. It means that both of the interacting partners exert an effect on the other genes. Typically, this occurs in convergently oriented genes. And we suppose that there is a third type of interaction, which we call as promoter competition model, which means that two divergently oriented genes overlap at their promoter regions, typically at the Tata boxes. Well, the thing is that our data refuse this hypothesis because this is not a competition. This is a joint control of the transcription, but I will show it a little bit later. So here is a complete set of the, let's say, subtypes of these interactions. I will go into details later when I discuss these cases individually. I only mention one thing before, that we can distinguish between the so-called hard and the soft interactions overlap. In the case of the hard overlap, there is let's say, a collision or interaction in any case. But in the case of soft overlap, there is an alternative. Normally, this is represented by alternative transcription initiation and transcription termination. So in one case, there is an interaction, and in another case, there is no interaction. That's why we will call it soft. The idea of the transcriptional interference is not a brand new idea. There were sporadic cases for pairwise interactions between viruses, bacteria, fungi, insects, and mammalian animals. What we claim is that this kind of interaction is not just a curiosity, but represents a widespread mechanism, not only in the viruses, but in the whole living organisms. So our model, what we use is an alpha herpes virus. This is a neurotropic herpes virus called pseudorabies virus. Pseudorabies virus is a popular herpes virus for the investigation of the pathomechanism of herpes viruses. It is also a widespread tool for the transneural tract tracing of neural circuits, and also as a vector for delivering foreign genes into neurons or cardiovascular tissues, etc., etc. So we use this virus for showing that the transcription interference is a really global mechanism. First of all, we sequenced this virus using the PacBio RS2 platform, and I'll show you a typical genomic organization of this virus. So there are genes in a tandem orientation, which is opposed with a similar cluster of the genes, And this genetic module is surrounded by divergent genes. And we can find every kind of interaction, every kind of overlappings between these genes. So this kind of genetic architecture calls for an explanation. That's why we try to investigate this issue. So first we determine the transcriptome of the virus using Illumina sequencing, both Illumina sequencing and the PacBio RS2 platform. And especially with this latter technique, we could detect several new transcripts, which was never been determined before. And this was allowed by the special properties of the PacBio sequencing. So we could detect very long transcript, typically polycistronic transcript. So in this virus, there are many different kinds of polycistronic transcription unit, but as it is known, the prokaryotic organisms contain this kind of units. Here, this is a little bit different because only the most upstream genes are very likely translated. The others do nothing. And we also could detect, for example, spliced and alternatively spliced transcripts, etc., etc. So let's start with the discussion of transcription overlap of the tandem genes. So actually, there are two different kinds of overlap. One of them is a so-called tail-to-head overlap, and practically all of the genes standing in the parallel orientation overlap in this way. The second type of overlap is, we call it as multiple parallel overlap, when the transcript share a common poly-A signals and also poly-A tails. Okay, and we can, I can show you here that the second case is most frequent here. So here are the polycistronic RNA, and the common poly-A has a largest peak. Here you can see that case. But we have shown that almost all transcriptional units have their own poly-A signals and poly-A tails. Many of them are represented only in a very few copies. Or there are also other genes. You can see here polycistronic genes which can be transcribed individually, having their own poly-A tails. This was sequenced using a poly-A-seq platform. So here I can show you the poly-A signals of the upstream genes. There are the major poly-A signals indicated by red or a blue arrow, depending on the orientation. And also there are rare, weak poly-A signals. The very weak poly-A signals are not indicated here. So I can show you the transcriptional dynamics of the virus. I will show you only some examples. For example, here we can see a cluster of genes. These are the USA 8, 9, and US2 genes. As you can see here, every combination occurs during the transcription. So the US2 genes can be transcribed individually. This is the same for the US9 and USA. And bicistronic and tricistronic products are also produced. So there's a special kind of dynamics. At the first hour, as you can see, the US8, US9 bicistronic transcripts are produced in a large amount, and the US2 in a smaller amount. Later on, at 4 hours, the US9 appears, and the proportion between the transcripts will be changed. The US2 will be transcribed in the highest amount. And this tendency is reversed once and then reversed back. So there is a special kind of transcriptional dynamics. So what we claimed, that the transcriptions interfere with each other. So it means when one transcript is, when one gene is very active, transcriptionally active, then it will suppress the other neighboring genes in every kind of orientation, the convergent genes, the divergent genes, and also the tandem genes. So when we analyze the transcript, and this is allowed by using the PacBio platform, because using PacBio we can discriminate between the different transcripts, including the polycistronic and monocistronic genes. We can see a special tendency when one gene is expressed. I mean, this is the, well, this is a value, our delta value. This is actually, there is the sequencing reads. It is called Roy reads in the PacBio sequencing. And we use the delta values, which means that we did use the R values at time t from the t minus 1 values. And we obtained this kind of data that there is an inverse relationship between the expressions. So this is the aim of our work, to find a relationship between the expressions. So here we can see the so-called hard overlaps, the hard convergent overlaps. You can see here this case, the two opposing genes means that. So this is exemplified with the case of the U30 and 31. So the heads of the two genes overlap with each other, and you can see here that there are the messenger RNAs and also antisense RNAs. And there are poly-A signals for both of the genes. We showed that the transcription does not stop at the poly-A signals, but with a certain efficiency, there is a transcription read-through. And later on, both the sense and the antisense DNA strand is active. So what could be the explanation for that? What is the role for these antisense RNAs? This is the basic questions. So if we examine the transcription using real-time RT-PCR, then we also find an inverse relationship. So if the messenger RNA, has a tendency up, then there will be a reverse tendency, and vice versa. So this is the analysis using a real-time PCR. We can repeat it using the PacBio platform, and we obtain the same. So we can see an almost completely inverse pattern of expression between the two convergent genes. You can see that the Pearson correlation coefficient is minus 0.55. This is very good inverse correlation, but not perfect. This is because we should examine, the transcriptional activity, but we only can examine the transcripts themselves. The transcript has a certain, half-lives. Maybe at a certain times there's no transcription activity at all, but there are transcripts from the earlier times remained in the cell. So we also can examine the effect of the joint effect of the tandem. Let's consider the UL31 genes. So there are two effects on these genes. One of them is the tandem partner, the UL32, and also the convergent partner. We can join their effect, and then if you see this picture, you can see also an inverse correlation. So if these two genes are active, then the middle one is repressed, and vice versa. The reverse is true if the UL31 starts to be active. So this is what I mentioned at the beginning of my lecture, that we supposed that there's a competition between the promoters at the overlapping regions. What we obtained, that in this situation there's not a competition, because if we consider the UL30 genes and compare the expression with the UL29, then we will have a same expression pattern. So it means that if there is a Tara box overlap, then this represents not a competition, but a joint coordinated effect. So we have a hypothesis. Two major points was proved to be good, the third one not. Okay, then we can also see such kind of, this is only just an example. Here you can see that UL9 and UL8 genes, if one of the genes are active, then the other genes are not so active, and then there's a change in the expression pattern. Well, you can see, for example, an alternative. We can see that there are alternative transcription terminations. For example, this is the US31, US35, US36. Normally, so these genes stop be transcribed here, but in some cases and at some time points, there is a termination which overlaps with the US36 genes. Okay, thank you. So here you can see that there are convergent overlaps in this indicated areas. This pictures was prepared 2 months ago. Today we determined more interactions, more overlaps. So soft overlap again. We can skip this, or maybe not. So I mentioned that with the case of the UL30 and UL31, there's a hard overlap. But the transcription continues across the polyase signals, and it produces antisense RNAs at the complementary DNA strand. This is the soft form of the interactions. If we examine this case with real-time PCR this is Illumina sequencing, then we can see that there are antisense RNAs with a relatively large amount. These antisense RNAs are produced by the transcriptional read-through mechanism. Finished. Okay, then I would like to thank your attention, and this is the acknowledgement pass. This is my groups who are here in Orlando. I would like to thank to the UD Genomed and Mark Snyder at the Stanford University Center. Thank you very much again for your attention.
We have time for a very, very short question.
Sure, we got time.
Okay.
Any big questions?
All right, thank you. Thank you. Let's thank our speaker.
Hi, yes, very intriguing talk, thank you. Since you can go ahead and crunch the promoter overlaps and transcriptional direction for genes in a known reference, and we have lots of transcriptomics data for those references, what would explain expression for genes that are transcriptionally close structurally that don't fit this hypothesis, where transcription of one hinders another and vice versa, but in real transcriptomics expression data, we don't see that kind of inhibition or consequential impact.
So the question is, if I could understand well, what is the mechanism for the interaction?
Well, if we see in observed data that there's plenty of now,
yes,
we don't see consequential impact of expression for one gene on another.
Yes.
When there's overlapping or intersecting promoters or they're close structurally. What is then the explanation of your hypothesis with respect to that observation? If we don't see that inhibition, and yet they're structurally, physically close, or they have intersecting promoters, or one by your hypothesis would hinder another, then what if we don't see that?
So you don't. So we have a large set of data. We could not examine all of the data now, but those examples, what we have examined, we could find the relationships, what we expected, at least in the case of the convergent and the parallel reoriented genes, and we could not find the relationship between the tether-box interaction. But if the overlap is larger, because in this virus there are 13 divergent genes, and only 2 of them exhibit tether-box overlap, the remaining 11 exhibit a larger extent of overlap, and these genes show the inverse pattern. But we show only the relationship between the gene expression. We did not show any, direct evidence how this interaction occur.